the semester, let's hope. Okay, so today, maybe we will finish this set of notes. We'll see, almost at least. So where we were last time was that we were just talking about this whole product concentration. It is recording, I think. Yeah, where we're talking about the whole product concentration thing with reference to enzymes. Do you remember that? We were kind of rushed at the end, so we'll just like revisit it, especially since it's been 5 days. So we were like looking at how to predict what certain things about an enzyme catalyzed chemical reaction would do to the rate of the reaction. And the last one that we were looking at was product concentration. I'm sorry, I sound like a dude today, but I've been battling the same cold. I told my husband it was sexy, and he's like, And he's like, no, you sound like one of the Marge Simpson's sisters. I'm like, thanks, babe. It keeps me humble. Okay, so this is what we were doing last time. I don't remember if it was exactly this, but we were looking at A plus B uses enzyme 1 to go to C, which dissociates to D plus E, which goes to the final product, which was F. And there were 3 enzymes involved. Right? Okay, so what we said was we were looking at enzymes based on like shape, right? And statistical probability, and so we were sort of understanding what the enzymes do in that context. And we said that enzyme 1 had this shape, and A was shaped like that, and B was shaped like that, or something, right? What are these shapes called, right? These sites called right here on the enzyme. Yeah, those are called the active sites, and this is enzyme 1. Okay, but remember last time we were saying that you can have another site. It must not be the active site, okay? Or else this, that would be a substrate. Okay, so you can have another site that is not the active site, that's not directly touching the active site, at which another thing binds. Let's say F actually binds there. The principles of negative feedback tells you that the product F, as the product of F increases, what does it do to the activity of enzyme 1? Increase or decrease it? It decreases it. Yes, it decreases it. And what was the objective there? So if, for instance, I wanted F, right? This is the concentration of F over time. This is the product that we were looking at. And the concentration of F over time, the ideal range is about there, right? Do you see that this little negative feedback inhibition mechanism would help me accomplish that? Because just look at it as statistics and probability. For instance, as your concentration of F starts to go up, then F actually binds, right? More of F starts to bind, which changes the binding site temporarily so that A and B don't fit, right? Does that make sense? Which means that that would cause the concentration of F to start going down, right? But as you had less F, right, more enzyme 1 would be active, right, which would make the concentration of F go up. That makes sense? As it starts to go up again, it inactivates more of enzyme 1, which makes it start to go down. Do you see that this mechanism is a little cellular example of negative feedback inhibition, right? And what would it do? It's serving to keep F in a homeostatic range. Does that make sense to you guys? Okay. So as the product concentration increases, and we don't need an abundance of product, we need a homeostatic range of product, then the product actually inhibits enzyme number 1. So concept check question that we talked about last time, but we were in a rush. So let's revisit it. Why does F not inhibit enzyme 2 or enzyme 3 as frequently as it inhibits enzyme 1? Any idea?
That's a waste of energy.
It's a waste of energy, it's a waste of intermediate products, right? It's a waste of materials, that makes sense? Now I'm not saying it doesn't ever inhibit more than one enzyme, but it usually feeds back toward the beginning so that you can stop the production of all of those intermediate products. Some of those reactions would be energy required, right? And so you could save energy and save intermediate molecules.
So you were saying if the concentration of F goes up,
Yes.
it will be more likely to fit in?
No, not more likely. It's more likely to find the enzymes, right? As you have more F, aren't they more likely to encounter enzyme 1? Yes, especially if it's intracellular. You're talking about an intracellular process in which F is produced and there are enzymes present, right? Just statistics and probability. If there is more F, it's more likely to find the binding site, right? And it's a loose and reversible binding site. Okay, does that make sense to you guys? Okay, so this is an example of what we're about to talk about. So this is an example of what we are about to talk about, and that is allosteric regulation. Okay, so conceptually, inside a cell, if you needed more enzyme, just make it, right? And if you don't need it, just don't make it. But do you think that that is going to be an effective mechanism for having the enzyme around when you need it? Is there any problem with protein synthesis, just making something when you need it? Is protein synthesis an instantaneous process? No, it's not. You have to go through transcription, and then you have to go through translation, right? And then you have to export it from the ribosome where they roughly are to the Golgi apparatus for processing and packaging, right? So this is not an instantaneous process. So not surprisingly, there are mechanisms by which you can turn on enzymes and turn off enzymes. So you can have them around, and if you use them all the time, you leave them on most of the time, and every once in a while you need to turn them off, you turn them off. But there are other enzymes that you rarely need, but you need them quickly when you need them, right? So you leave them around in the off position and you can turn them on. Does that make sense? Okay, so these regulatory mechanisms are mechanisms by which you can turn on and off enzymes, and they are called allosteric regulation and covalent regulation. So let's visit allosteric regulation first. Allosteric regulation is like this top figure right here, just what we were talking about. You can loosely and reversibly bind something at any site other than the active site, which we generally call a what site? We talked about it last time. We call it a regulatory site. You loosely and reversibly bind it there. By loosely and reversibly, I mean it's more likely to be a hydrogen bond or an ionic bond than a covalent bond. So those are quicker to form and quicker to break, right? Okay. Loosely and reversibly, and by binding to the regulatory site, you can temporarily change the shape of the active site. Because remember, this is one folded protein, right? And you can like squish on something in one place and it can make a bump pop out on the other place, right? Okay, so this is called allosteric regulation, and allosteric just means a shape change. And so I have this thing called a modulator, and modulator is really a general term. It could be anything other than the substrate, right? Anything other than the substrate. I'll give you some examples in just a second. So what they are going to do is the modulator can temporarily bind and turn it on or turn it off. This is how a common enzyme works, right? So as the substrate concentration increases, the reaction rate increases until you hit saturation, right? This is how something that actually needs to be turned on works, right? It takes a little longer because you have to wait for the modulators to bind. Does that make sense? Okay. But it also means that you can individually, per one enzyme, you can turn that enzyme on and off, right? But can you see collectively, per that whole pool of enzymes, you can turn the enzyme activity up and down, right? If you have more modulator and you're using the modulator to turn it on, if you dump a whole bunch of it, you get a really fast rate at which you hit saturation, right? Dump a little bit of it and you get a little bit faster, right? So individually, you can turn one enzyme on and off, but collectively, you can like use it as a volume knob, right? That modulator, how much modulator are you going to put in? That makes sense? So they can either be default off and you turn them on, or default on and you turn them off. Did you have a question? Okay. So how is this related to your human body? Because you know, but people never know, and I didn't either, why you need certain things that are not really directly involved in your anabolic or catabolic processes. So we know that we need in our diet, you need these nutrients, like you need fats and sugars and proteins and those kinds of things. And you know that water is involved in a bunch of different processes. But you also know that you need vitamins and minerals, but nobody ever tells you why the hell you need them. And you don't need them in large quantities. For instance, you're not using them for catabolic processes. You're not taking vitamin D and breaking it down to make ATP. And generally speaking, you're not using vitamin D as a large component of something you're making as a structural component of your body, right? Does that make sense? So how the hell do you use these? Quite often, interestingly, your vitamins and minerals, which are only needed in trace amounts, are used to activate and deactivate enzymatic processes. And so if they are not present, does it mean that you never actually get the product, or does it mean that you get the product far less efficiently? It generally means that you get the product far less efficiently. So they are called cofactors or coenzymes, and what they generally are is modulators for enzymes. Now, sometimes what they do, interestingly, is let's say I have a biochemical pathway that's 26 steps, right? All of them requiring enzymes. You guys with me? Okay. Sometimes I know that step number 25 is going to require copper, right? And that copper is rare. It's hard to get in the body. It's actually a mineral that we need in the body. So does it make sense to you that sometimes in step 1, 2, or 3, I will use copper to make sure it's worth going to step 26? I'll use copper as a cofactor or a coenzyme to say it's okay, you can keep going, right? Because there's copper. Does that make sense? If it's a trace mineral that I don't have in large quantities, can you see why I might want to check early in the process before I commit all of these intermediate molecules? So you will see vitamins and minerals. Minerals are generally inorganic. They are inorganic. They're usually metals or something like that. You can get them by licking a rock as long as you get the right rock. Calcium and magnesium and copper and zinc and those kinds of things are minerals. You need them in trace quantities. The difference between a vitamin and a mineral is a vitamin is organic, which means you could get it by eating a plant or a puppy. Don't eat puppies. But it has to come from another living thing, right? That's still needed in trace quantities and not directly used as anabolic or catabolic processes. Does that make sense? So those are the modulators. So they're less likely to be substrates and they're more likely to be something that turns on or off an enzyme. Does that make sense? Okay, so that process that we just talked about was allosteric regulation. And so I want you to look at allosteric regulation and notice that really the objective is to temporarily change the shape of the active site, right? And now I'm going to show you another way to do the same thing. The other way to do the same thing is called covalent regulation. Please note that you are accomplishing the same thing. Active site is temporarily changing shapes. But this time you are going to be using covalent bonding instead of the weaker types of bonding, which means, do you imagine that it's going to be faster or slower than allosteric? Slower. More or less likely to require energy. It's more likely to require energy. In addition, you're also going to need helpers. So objectively, what I'm going to try to do is try to get this enzyme to change the shape of its active site and therefore its activity, right? But please note that it's going to need 2 other enzymes to help it, okay? It's going to need one to form that covalent bond, and then when I want to undo it, I'm going to need another enzyme to undo that covalent bond. The objective is the same, okay? So you have run into these before if you've ever seen kinases and phosphatases. Have you ever seen a kinase or a phosphatase? A kinase is a common enzyme that actually phosphorylates another protein or enzyme. So in your notes, it says basically you take an enzyme and you add an inorganic phosphate and you get a phosphorylated enzyme. And that's called a kinase. So you guys tell me which one, enzyme A or B, is a kinase if the red group is a phosphate. A or B is a kinase. A. Yeah, A is phosphorylating. Does that make sense? Okay. Now, phosphates are really common groups that we use for covalent regulation because we have a lot of them around. Where do you think that is the go-to place that we will snatch a phosphate from? ATP, yes. We very commonly snatch a phosphate, slap it on something, it changes the activity, and then when we want the activity to go back, we pull the phosphate back off. So we use this next enzyme, which is a phosphatase. Okay, and the phosphatase snaps it back off, right, and changes it back to its original activity, right? So you're going to need 2 buddies and generally some kind of ATP. Not always, but some kind of ATP. That makes sense? But the objective is the same. These are just 2 different ways of turning enzymes on and off. Covalent regulation and allosteric regulation. Everybody good? Okay, so keep that on in mind. And then we will move on to the concept of metabolic pathways, which we've actually already been talking about. Okay, so what is a metabolic pathway? Like I said before, generally speaking, if you want a complex product inside a cell, it doesn't happen by a single chemical reaction. It happens by a series of enzyme-catalyzed chemical reactions, and that is a metabolic pathway. And the metabolic pathways get ugly and hairy. I'll show you just one. Actually, I'll show you a few. But this is not how you are going to learn them now. Although if you take biochemistry or organic chemistry, you have to draw all the damn molecules and know the enzymes. Yes. Something to look forward to. That's not what we're going to do in here, but let me show you this just so you can get an appreciation. What this is showing you is this is just the first step of cellular respiration. This is just glycolysis. It's a step of 10 enzyme catalyzed reactions. Please look. Each enzyme, hexokinase, phosphoglucoisomerase, all the way to pyruvate kinase, each one is catalyzing a separate chemical reaction, okay? Now, what we're going to do is instead of looking at the individual components of a biochemical pathway, we're going to look at a really important biochemical pathway, and we're going to focus on, instead of memorizing the individual components, we're going to focus on application to human physiology, okay? So, and of course we are going to do cellular respiration, and I know you probably hate it as much as I hated it when I was learning it as an undergrad. I resisted learning cellular respiration 5 times when they tried to teach me. I would just memorize it and then do a brain dump, and then memorize it and do a brain dump. My objective with you guys is to make it relevant enough that you will actually remember what it has to do with anything. Sometimes I succeed with 2 of you in class, and the rest of you just continue to hate it. But I am forever an idealist, so I'm gonna give it a shot. Okay, so the metabolic pathway that we are going to focus on is going to be cellular respiration. Cellular respiration is a series of enzyme catalyzed reactions. It starts here. This is the first step of cellular respiration, which is actually 10 steps. This is the second step of cellular respiration, which is Krebs cycle. And then the electron transport chain or oxidative phosphorylation is the third step. But that's not how we're going to approach it. What we're going to do is we are going to try to understand how it's relevant. First thing is that you are going to only have to keep track of the important players, which means that, of course, you have to keep track of the ATP, but you don't need to keep track of the NAD or NADP or FAD, right? You're going to keep track of the ATP because it's going to be directly relevant, because I will talk about how you will use the ATP that you just generated to, for instance, run your sodium potassium pumps in that cell, and what would happen if you didn't, okay? And you're going to have to keep track of the stuff that goes in the front end and out the back end, and not any of the intermediate stuff, right? Okay, so first thing is we're going to do aerobic cellular respiration, and we're going to do glucose. So glucose is definitely not the only thing that you can burn, right? You can burn other sugars for ATP. You can burn fats for ATP, and you can also burn proteins for ATP. Yes? Okay. But why is sugar so damn important? Because it's the only thing that the brain really likes. And if the brain ain't happy, ain't nobody happy. So it's really like the sexiest of all the molecules. It's the one that can always get into every cell or every club. Like, it's like the popular molecule. It's not the only one. So we'll do aerobic cellular respiration of glucose. So we're going to start out with C6H12O6, which is, of course, glucose. And ultimately, you guys, the reason that we are doing this is because we are trying to make this. Yes? That's the objective. So with that in mind, before I even go any further, am I talking about an anabolic process or a catabolic process here? I'm talking about a catabolic process. I'm going to take something relatively large, and I'm going to break it down ultimately, right, until I get ATP and some smaller molecules. But in order to do this effectively, I don't need glucose alone. In order to do this effectively, I actually need oxygen. And then it will break down to smaller molecules, right? Can we agree that these are all smaller individually than this one? Yeah? And you're going to produce ATP as a byproduct. So what we're going to do here is we're going to go through the 3 basic stages of cellular respiration. And we're going to refer back to this equation, and we're going to figure out where each of the reactants is used and where each of the products is generated, right? We're going to figure out what would happen in a cell if you didn't have oxygen. How much ATP would you be able to generate? How would that impact the physiology and the homeostasis of the cell, right? So we're going to try to make it relevant. The first way to make it relevant is just to acknowledge that this equation is the entire reason you eat, breathe, and have a cardiovascular system. The entire reason that you breathe. Let's start with breathing. Embarrassingly long time is how long it took me to figure out that the reason this damn thing is called cellular respiration. Because at the cellular level, You're taking in oxygen and giving off carbon dioxide. I was like, hello, Melanie. That was like 10 years, and then I had a light bulb moment. Has everybody known this? See, I just didn't try to learn it. I was like, I'm just going to write this down. I'm going to take it on the test. And I'm never going to know it. That'll show you. Literally showed nobody but me. So you're taking in oxygen, you're giving off carbon dioxide. This means, of course, in the absence of oxygen, what's going to happen to your level of ATP? Decrease, right? This also means that the faster that this occurs, what happens to your carbon dioxide concentration? Increases. If you were not blowing it off, is there any impact of accumulating carbon dioxide? We talked about one last time. Increasing carbon dioxide tends to change something that pH, yes, it changes your pH. So this is directly going to be related to the homeostasis of the whole body. The whole reason that you eat is because you need some large organic molecule that you can use to break down via catabolic process to generate ATP, right? And how about a cardiovascular system? This crap has to happen all the way from your big toe to your brain cell, right? And diffusion is not effective enough to transport these things from one place to the other. So the cardiovascular system is primarily about bringing around nutrients, waste products, and gases, right? So that you can do this in every single cell in your body, right? Okay, so this is the summary equation of aerobic cellular respiration, okay? The only thing that we're going to keep track of numbers on is ATP, and you do not have to balance it. As a matter of fact, don't try to balance it unless you're very confident, because you'll probably get it wrong. So be careful. Okay, so let's talk about the stages of cellular respiration. Okay, so instead of looking at it in that kind of detail, let's do this kind of detail. Okay, this looks terrible, but it's not. This is a summary of the entire process. Please note that ultimately what you're doing is putting in glucose and getting out ATP, yes? Anything that comes out on this side is going to be a byproduct, right? And stuff that goes on this side you're going to need. They left off one oxygen, but we'll just deal with that. Okay, so there are basically three subsets of chemical reactions that are involved in this whole process. This is just keeping track of the sum of what goes in and the sum of what goes out. But there are three processes. There's glycolysis, okay? Looking at the name of glycolysis, what kinds of things do you think you generally break down in glycolysis? Sugars, fats, or proteins? Sugars, yes. We will see that you can also burn fats and proteins. We'll talk about that. Glycolysis, this intermediate stage is called the Krebs cycle. We'll talk about that. And then this last stage is, depending on how you learned, either called the electron transport chain or oxidative phosphorylation. Note to self before we go further, you're probably in the habit of, from cell biology or 1610, thinking that the organelle in which cellular respiration occurs is what? Mitochondria, and that's true-ish. But it's not completely true, because glycolysis is actually older than mitochondria. Glycolysis is actually older than eukaryotic organisms. It occurs in prokaryotic organisms. So it actually occurs in the cytoplasm or the cytosol, the intracellular fluid. It occurs outside of the mitochondria. These two stages occur in the mitochondria. Anything that occurs in the mitochondria requires oxygen. Okay? So we will see that glycolysis does not. It occurred actually before oxygen existed in high quantities on the planet. So it predates high levels of oxygen on this planet. And then these two are mitochondria, occur in the mitochondria, and they require oxygen. So let's start with glycolysis. So glycolysis is actually a series of 10 enzyme catalyzed reactions. And the name tells you what it's doing. It's breaking down sugars. And really what it's doing, if you start with glucose, it takes glucose, which is a 6-carbon sugar, and it pops it in half to 2 of these pyruvates or pyruvic acids, which, if you care, each of them is a 3-carbon sugar. So it almost just pops it in half. That makes sense? Okay, and then there's this little reaction that occurs right in here. Biochemists would hate that I'm lumping this in with Krebs. but I'm going to lump this one in with Krebs. What has to happen is you lose a carbon dioxide and then it's ready to go into Krebs. This is called the linking reaction. I'm just going to lump it in with Krebs. So it's Krebs and the linking reaction. So let's talk about glycolysis a little bit more before we leave that. Okay, so it occurs in the cytoplasm or cytosol. It doesn't require oxygen, so it can and will occur in anaerobic conditions. That ends up becoming important. If you are using your skeletal muscle for lifting weights, right, and you do that thing that people do, which is like start holding your breath to the point, so you're going to run out of ATP really quickly because we will see that glycolysis does not produce an impressive amount of ATP at all, but it can and will do it in the absence of oxygen. So it doesn't require oxygen, can occur in anaerobic conditions. And so if oxygen is present, what's going to happen is you're going to keep going. If oxygen is not present, the best you can hope is that some cells can send it right back through glycolysis backward, which will generate a little bit more ATP, but will also generate potentially lactic acid as a byproduct, which tends to fatigue muscles. And we'll talk about that in skeletal muscle chapter. So if oxygen is present, you get to keep going. If oxygen is not present, very little ATP is formed. Lactic acid may ultimately be formed as a byproduct. So what we're going to do is we're going to keep track of where each of these is used and where each of these is generated. Ready? So, of course, did we use glucose in glycolysis? Yes. Did we use any oxygen? No, we did not. Did we generate any carbon dioxide? Not in glycolysis, we generated after in the linking reaction in Krebs. Any water? Got it up there if it's used. And any ATP? Yes, we did. What happens is you actually use 2 ATPs and generate 4. So if you just keep track of the net, what happens is 1 round through glycolysis with 1 molecule of glucose will generate 2. Does that make sense? Good? Okay. So let's say that we are rich in oxygen and we're going to keep going, right? Because 2 ATP is not impressive at all from a single molecule of glucose. It literally runs 1 ATP sodium-potassium pump 2 times, right? And you have sodium-potassium pumps on every damn cell, right? So you need a hell of a lot more than 2 ATPs per glucose molecule. So if we have oxygen, we are going to continue, right? And we're going to continue into Krebs and then hopefully oxphos. So Krebs. So this is called the linking reaction. I don't care that you memorize it separately. What it does is takes pyruvate, pops a carbon dioxide off of it, and makes it into this acetyl coenzyme A, which is interesting because we just talked about coenzymes, right? They actually fit into another enzyme and modulate their activity, and that's part of what that one does. So acetyl coenzyme A, that's the linking reaction, and then you push it through Krebs and notice what's generated in Krebs. You potentially generate some more ATP and you also generate carbon dioxide. So notice that all of the carbon dioxide from the entire process is generated in this middle step, right? And this middle step doesn't even always produce ATP. It does produce this byproduct that can become problematic though. So it occurs in the mitochondria, and everything that occurs in the mitochondria requires oxygen. And it produces about 2 ATP. Now, why do I say about? Because there's actually 2 different ways that you can actually make this occur. One way that you do it, you produce 0 ATP, and the other you produce 2 ATP. But the important thing is that they both end up with this coenzyme 2H that allows you to go through the impressive part, which is oxidative phosphorylation. So let's go back. Did we use any more glucose? We didn't use more glucose, right? We're still working on the same one. Did we use oxygen? Yeah, so we used oxygen for the first time in Krebs. Did we generate carbon dioxide? Yeah. Did we generate water? Did we generate water? Water is generated from the last part, right? Okay, so we didn't generate any water yet. Did we generate ATP? Potentially. So the best we can say about Krebs is that it either generates 0 or 2. Okay? And it doesn't really matter because that's not the important part. The important part is the next part, which is oxidative phosphorylation. If it wasn't already obvious, something that has the word oxidative in it is generally going to require oxygen. Okay? So oxidative phosphorylation is next. So why bother with this? The reason we bother with this is because we can't get from there to there without going through linking reaction in Krebs. So Krebs is really just about getting from point A to point B so that you can actually do the impressive part, which is oxidative phosphorylation. Okay, so oxidative phosphorylation is where if you're a carb, you're going to continue as long as you have oxygen. It's going to occur in the mitochondria, so it requires oxygen, exactly. It produces water as a byproduct. This is called metabolic water, and nobody really cares about it because it just mixes in with the other 60% of water that you have in your body. And the impressive part is that from a single molecule of glucose, one time through, it produces 34 ATPs. So that's why we're trying to get there, and that's what oxygen does for you. Okay, so one more time. Did I use any more glucose? Did I use more oxygen? Yes. Did I generate more carbon dioxide? No, I didn't generate any more carbon dioxide. I actually generated water. And how about ATP? So oxphos generates 34. Okay, so this means that from a single molecule of glucose, in the absence of oxygen, how many ATPs are you going to generate? 2. In the presence of oxygen, you are going to generate what? 36, 38. Yeah? That makes sense? The hell do you do with those? You use them for all of your, for instance, anabolic processes like protein synthesis, right? You use them to maintain homeostasis. You use them to run your sodium potassium pumps. You use them to regulate the cellular pH. You use them to make sure that you have the proper concentrations of ions on one side to the other, right? That makes sense? You're going to use one of these to start the next round of this, right? Because remember, it uses 2 and generates 4. So you're basically going to be using those above and beyond all things to maintain homeostasis. And then after homeostasis, if you have maintained it, you can start to do extra things like growth, like repair, like protein synthesis, right? Which one do you think gets dibs on your ATP? Homeostasis or protein synthesis? Homeostasis gets dibs. And then later, if you have extra, you can do protein synthesis. Does that make sense? Okay, so ultimately what you are going to have to do by the time your first test rolls around is there are very few chemical reactions that you're going to have to know for me. This is one, you've already learned the other one, right? The pH one. So you're going to have to be able to write for me the unbalanced summary equation for aerobic cellular respiration, right? You have to be able to tell me where things are used or where things are generated. And then you're going to work on this one on your own. You're going to make up in 3 sentences or less how this is relevant to human physiology. And so what you should be doing before the first test is listening to see how ATP is used and what it has to do with human physiology, right? And then you start putting together an answer that has content in it. Don't, for instance, say you would die without it, because you knew that before you got here, right?
Did you get that?
Oh, all the time. All the time. You die without ATP. I'm like, well, apparently you don't need me to tell you anything about ATP. So ideally, you would say something about homeostasis. You might put in a pump. You might tell me something about active transport once we get to it. Start telling me something about what cells do with ATP. Good? So you can work on that until the first test. Okay. So now, let's back up just a little bit and let's talk about the fact that you don't just burn glucose, right? You burn complex carbohydrates and simple carbohydrates. So, what's complex carbohydrate? Well, a simple sugar is a sugar that can be put directly into this process, and it's not only glucose, it can be other simple sugars. And a complex carbohydrate is basically when you take a bunch of simple sugars and you link them. So what would I do with any complex carbohydrate before I would put it into glycolysis? I would break it down to a simple one, right? And then that simple one would go into glycolysis. So let's put this together with the bigger picture because we talked a little bit about diabetes and insulin and glucagon and that kind of stuff before. So in order to start this process, you have to get into the intracellular fluid, right? So the glucose has to go from the ECF to the ICF in order to do any of this, right? The situation occurs in which you have your swimming in sugar, right? But the cell can't take the sugar in, because a lot of times what occurs is the key to unlock the door for the sugar to come in is insulin, right? So we can talk repetitively throughout this entire semester about the ability of an untreated type 1 diabetic to maintain cellular homeostasis in something that's incredibly energetically expensive, like a neuron. Can you see how you can have trouble maintaining homeostasis with an untreated type 1 diabetic? What would they not have? What would they not have in order to do this process? They wouldn't have the glucose. They would run out of ATP, and you lose the ability to maintain cellular homeostasis. Especially big deal when you have a cell that, if it gets to the point that it can maintain homeostasis, dies and is amitotic, like a neuron. Do you know, have you ever heard of diabetic neuropathy? In which a diabetic has the sort of progressive deterioration of their neurons that reduces sensation and those kinds of things. This is directly related to that story. If you have a cell that can't maintain homeostasis and eventually just dies, and the cell is also amitotic, then those damages are permanent. Does that make sense to you? Okay, so this is directly related to the story that we're talking about. So what happens is glucose generally has to enter the cell by way of like facilitated diffusion, which is a transport mechanism we'll talk about again in the next set of notes, in the presence of insulin. Not every cell in the body needs insulin to bring glucose in, but most cells do. And then we know what happens to glucose after you get it in there. It gets broken down to pyruvate or pyruvic acid, and then ultimately it gets broken down to ATP. But that means that glucose is pretty damn important. If also I remind you that glucose is your brain's favorite nutrient, your brain's favorite thing to burn by catabolism. And by favorite, I mean that up until 5 years ago, they thought that the brain could only burn glucose. And now we know that it preferentially burns glucose. And if you run out of glucose, your brain is definitely going to let you know about it. It doesn't switch nutrients very quickly. So that means that glucose is pretty damn important. So whenever I take in a ton of it, like at Halloween, I don't just pee out the extra. I better store it, right? So there's a storage mechanism for glucose, and there's a retrieval mechanism for glucose. So let's talk about that. So if your brain really, really depends on glucose, then I have to have a way to make a glucose savings account that I can always make withdrawals from. This is happening in you every day, okay? So here's what we already know. And there's going to be a lot of terminology that you're going to use here. So if I take glucose and I break it down ultimately to ATP, it's not going to break down immediately to ATP. This process starts with glycolysis. Everybody okay with that? Starts with glycolysis. Okay. And what I want to do is I want to show you the similarities between this terminology. Okay. So I took in glucose, I broke it down to ATP. I used the ATP for homeostasis, for instance. But it's the day after Halloween, so I took in an excessive amount of glucose, right? And then I'm not always going to be doing that. There's always periods in between meals in which you're not eating, but your brain gobbles up about the same amount of glucose sort of constantly. So it always needs to be able to sort of be fed with the same amount of glucose. So what happens is, when times are rich and you have an abundance of glucose, you always want to make deposits into your glucose savings account. So the deposits that you make into your glucose savings account, you save as, does anybody know how animals store glucose? Anybody know? It's primarily in your liver and your skeletal muscles, and you store it as glycogen. Does that sound familiar? If you were a plant, you would store glucose as starch, but you're not a plant. Good? Okay, so what I want to do is show you what this term is called. So you're going to store that as glycogen in your liver. It's the day after or the moments after you've gorged yourself on Halloween candy. And you're going to end up peeing out some of that glucose, just so you know. But you're going to use everything you can, and then you're going to try to store as much as possible before it gets lost to the kidneys. You're going to store it in your liver and your skeletal muscles. And this process is called Glycogenesis. I'll break it down. Genesis, glycogen. What does that mean?
The creation of
Exactly, the creation of glycogen, right? All of these terms are going to be very similar. So I'm teaching you how to break them down because memorization is not going to serve you. And I'll put all 4 of them on every question about any of these terms. So this is glycogenesis. Okay, concept check question for you. I don't expect to get you to get this, but I want you to hazard a guess to see if you can figure it out. There is a hormone that is a trigger for glycogenesis. I'm going to give you 2 choices. You choose which hormone. You don't have to know this. I want you to logic your way to it, even if it's uncomfortable. Which of these 2 hormones do you think is a likely trigger for glycogenesis? Insulin or glucagon?
glucagon. Insulin. No, insulin.
Just think about it for a second. Don't wait for the other yahoos. Nobody knows. Don't wait for them. They don't know either. Which of these 2 hormones is likely to be a trigger for glycogenesis? Insulin or glucagon? I'll explain it to you, and I won't test you over this. I just like to see whether you can start getting the big picture before it's actually here for you. Anybody relatively confident about their answer? I'll be nice, I promise.
Yeah.
Go ahead.
Well, glucagon, because that's when glucose is gone.
But glucose isn't gone. Glucose is abundant. It's insulin. Yes. When do you want to make a deposit into your savings account? When you're rich, right? So I'm rich in glucose. I make a deposit. So your deposit slip is going to be insulin. Does that make sense? I bet you guys will be able to figure out how you write a check in just a second. Does that make sense to you guys now? Not did you get it the first time, because people don't, and I didn't expect you to, but it is true. Okay? Does that make sense to you guys now? So when times are rich, in addition to like having all your cells gobble up your glucose, this is another way to drop your glucose level, right? Like putting it in the bank, right? That makes sense? Insulin's primary objective is to drop your blood plasma glucose level. It does a lot of it by sticking it inside the cells, and then once all of the cells are flush, you stick it in the bank, right? Okay, so making a deposit into savings doesn't do anybody a damn bit of good unless you can make a withdrawal when times are tight, right? So you are going to make a withdrawal. And this withdrawal is, look at the word, taking glycogen and making it back into glucose so that you can burn it, especially for the brain. This term is Glycogenolysis. Do you see the term? You actually say Glycogenolysis is how you say it. But I want you to look at it. And please note that it's really easy to confuse with this one, right? This is glycolysis, which is the breakdown of sugars.
That's the creation and breakdown.
This is glycogenolysis, which is the breakdown of what? Glycogen to glucose, right? Does that make sense? Okay, everybody good with that? Okay, what?
The breakdown of glycogen to
The
glucose.
breakdown of glycogen to glucose. Does that make sense? Okay, so I know you know now, but now hazard a guess as to which of these 2 hormones is the trigger for glycogenolysis, insulin or glucagon? Glucagon, because you need it when your glucose is gone, right? Have you ever had those moments? I know you have. If you're busy, and I know everyone's busy, right? You just forget to eat for so damn long, and then like once you have time to eat, you don't have anything to eat. Like I don't have time to eat. Eating and peeing during the workday is like impossible for me to figure out how to accomplish. I'm like I hate having to pee during the day. So, but here, have you ever also felt this when you're like so damn hungry that you're dumb and mean and you can't figure out like how to work a box? What? And then and then all of a sudden, 10 minutes later, you're fine. You're like, oh, I got this. I'm fine. You know what just happened? This. You just dump some sugar into your bloodstream and your brain's like, we can do this. You are bigger than that box. You can manage the box and the doorknob simultaneously. Right? Does that make sense? You just dump it into your bloodstream and then your brain's like, I can function again. That's glycogenolysis, which is triggered by glucagon. Glucagon happens in between your meals. Right? I release insulin right after I eat. and then all my cells gobble it up, and then eventually I'm like, okay, I'm out, right? I'm out. Brain still needs glucose, been hungry for a little while. Glucagon is released, dumps sugar into my bloodstream for my brain. That makes sense? Okay, and then one last thing. What happens when times are really, really tight and you have exhausted your savings account and you're not eating? Do you just say, it was a good run? No, you sure as hell don't. You start eating yourself, is what you do. Your brain does not play well with others. It is only concerned about itself. And if the brain ain't happy, ain't nobody happy. So what's going to happen when times are really, really tight? Like on Friday. I got the flu on Friday. I slept 18 hours on Friday, right? My brain still requires about the same amount of energy when I'm awake versus asleep. I was not eating anything, obviously. How the hell did I stay alive? How do you guys stay alive? How do people live through the first trimester of pregnancy when you can't eat a damn thing, right? This is how they live through the first trimester of pregnancy. They do this little process which says, okay, ran out of glucose, you're apparently not going to feed me. Times are tough. What I'm going to do is I am going to take amino acids, which I will get from where? Proteins. What do you think is a very, very accessible place for protein in your body?
Muscle.
Skeletal muscle. You start to break down your own skeletal muscle, and your brain can't burn amino acids directly. You have to take the amino acids and make them into glucose, which then your brain can burn. And you do this by a process, look at it broken down, called Gluconeogenesis. Neo, what does that mean? New. Genesis. What is it? Genesis. The beginning or the creation. So what are you creating? You're creating new glucose. Exactly. Yeah? That makes sense?
No, is this after 18 hours of events?
It depends
Or is
how
it
much you had stored.
10 days.
When people carb load, they're going to have a lot in their savings account, right? So you're not going to go into this very quickly. If you are, for instance, a person suffering from anorexia, you can imagine that not only would you not have much glucose in your system because you weren't eating much, you would have very little stored, correct? And then you would be very quick to flip into gluconeogenesis to keep your brain alive, right? This also happens with people who do this thing, which is, I really, really want to lose weight, so I'm not going to eat. I'm going to work out and try to build muscle mass. How's that going to work for you? You are actually going to be gobbling up your muscle mass to keep your brain alive, right? And the process of building happens after homeostasis, right? That's gravy after homeostasis. Does that make sense to you guys? Okay, so these processes are all directly related to one another, right? And you're all trying to get here. And the reason there's so many creative storage mechanisms for glucose specifically is because glucose is the nutrient that is preferred by the brain. And when the brain is, by the way, when the brain is really low on glucose, it tells the rest of the body that you don't get any. You burn something else. You burn fats, you burn a different sugar, or you burn proteins, but you don't get any glucose. So the brain increases its glucose uptake and tells everything else to suck it, right? I'm telling you, it's like, the brain is a total capitalist. Okay, so you guys all right with that? Those are relationships. Now, the terms, I can't memorize them, but I can always figure them out by context, right? And so what you need to do is to figure out with each one, you were taking what and making what into it, right? You were taking what and making what into it. Go ahead.
I mean, I don't want to say it wrong, but
Gluconeogenesis.
Are you, so how you said it's making new? I should make it
So what you're doing is this was an amino acid, and you're making it into glucose so that you can burn it by the brain. That's why it's new,
okay,
glucose. You didn't ingest it as a sugar, and it wasn't a sugar when it was part of your skeletal muscle. You can do this, by the way, if you're doing a super, super high protein diet too. If you're like damn near zero carbs, you are forcing this equation with stuff that you took in instead of your own skeletal muscle, right? We'll talk about the fact that it's a little risky. It's certainly functional, but it has some risks associated with it. We'll get to those in just a second. Okay, so now let's go back and revisit the idea that sugars are not the only thing that you burn, right? You can also burn fats, and you can burn proteins. When you burn fats, most of them skip glycolysis, except for this little guy called glycerol, which is a weird little fat sugar hybrid. Most of them skip glycolysis because they're not glycos, right? And they go in right before or right after Krebs, okay? What is significant about fats is that per gram, they have more ATP generated than gram of carbs or a gram of protein. So they are an effective storage vesicle. The process of breaking down lipids is called lipolysis. And lipids are, you can understand, because lipids basically what they are is they're like a really dense energy storage place. And if you're trying to store something, right, you get a small storage area, you stuff as much as you possibly can in there. So it's all well and good when you're trying to store it, but when you're trying to burn it, it's a bit more problematic because they have more energy available per gram than are the carbs or proteins, which are similar. So the liver is intimately involved in anything related to lipids. The liver has to make bile. If you eat something that's really fatty and you're not making any bile and dumping it from your liver to your gallbladder into your intestine, then you're going to poop like a goose. It's going to go straight through you, okay? People who have gallbladder surgery have to be very careful about the amounts of lipids they take in afterward. So you can't absorb lipids in the absence of good liver function. In addition, lipids are hard to carry from point A to point B because they're like the original ADD. Like everything is lipids and you're lipid soluble, right? So I'm trying to get from my intestine to like my little finger, but I'm lipid soluble and every single cell is made of lipids, right? So trying to get a lipid to go from point A to point B without escorting it is like trying to drop your toddler off at the end of the Walmart parking lot and ask him to meet you at the door. You know you're never going to see him again, right? Ever. So what has to happen is somebody has to hold their hand and make them temporarily insoluble. And those proteins that do that are made by the liver as well. So think you got liver problems, you got liver problems, basically. Okay, and then briefly let's talk about LDLs, VLDLs, cholesterol, the whole cholesterol angle. So big picture, short version is that dietary cholesterols, most of your dietary cholesterols are from animal products because cholesterols are only found in high quantities in animal cell membranes. You don't actually have to eat the animal. You can eat a product of the animal like an egg or milk from a cow. And there are good and bad cholesterols. It's easy to remember which one's which because the low ones are bad and the high ones are good. So you got LDL, which is low-density lipoprotein, and VLDL, which is very low-density lipoprotein. And then you got HDL, which is high-density lipoprotein. What's bad about the bad ones is that when they increase in concentration in your bloodstream, they tend to gunk up the sides of your blood vessels. When they do that, they narrow the lumen, which reduces blood flow, and they also increase your blood pressure, which is harder in the cardiovascular system, right? If you gunk up a whole bunch of vessels, right, and expect it to transport the same amount of fluid, then the pressure goes up. So what are some foods that you can eat that would pretty damn dependably increase your cholesterol level? Yeah, meat, especially the fatty meats, like your favorite, like bacon. Bacon is like a cholesterol bomb. What? Cheeseburgers, yes, with an egg on top, right? Yeah, everybody's like, oh, yeah. So yeah, they're pretty high cholesterol foods. Can you be a vegetarian and have high dietary bad cholesterol? Yeah, every vegetarian I've ever met eats a boatload of cheese if you give them the opportunity. Now, if you are a vegan with high LDL, then it means you, because you're an animal, are making a lot of LDL. Okay, it doesn't happen to very many of us that occurs. The HDL actually counteracts the LDL. High-density lipoprotein is a type of cholesterol that is hard to get in high quantities from your diet. You can get a little bit of it from your diet. What kinds of foods are pretty dependable for that? Anybody know? Some sorts of fish are pretty dependable. But what's the really good way to get high levels of HDL? Exercise. Sorry, the answer is always diet and exercise. I don't have any other answers for you. What happens with HDL is the amount of HDL that you have in your bloodstream contradicts the amount of LDL because it keeps the LDL from sticking to the walls of your blood vessels. So if you're going to In-N-Out Burger, you run there and run back, and then you feel good about yourself, right? Which literally nobody does. Okay, so The last thing that we will do, and we'll just barely talk about it today, is the fact that you can burn proteins as well. Please look. Proteins skip glycolysis, right? And they either go in before or during Krebs cycle. Last thing I want to tell you about burning proteins as catabolic processes is that it will cost you something to do that. So these super high protein, low carb diets, right? They do have some risks associated with them. One of the risks is this. One last thing before you guys leave. In order to burn a protein, catabolic processes, I can use proteins for anabolism too, right? I can use them for protein synthesis. But catabolism gets dibs, right? If you don't have enough for homeostasis, enough ATP, then you're not going to do any protein synthesis. So I'm going to take proteins, and in my stomach, I'll break them down to amino acids. And then if I'm going to do catabolism with them, the first thing I have to do is pop off the amine group of every amino acid. That creates a potential problem because this is ammonia. When you are using proteins for catabolic processes, you generate ammonia. You have to send ammonia to the liver to detoxify it to a less toxic substance called urea. And then you have to get rid of urea where? Kidneys, exactly. So these super, super high protein diets are potentially really hard on your liver and your kidneys. Like Atkins diet, that was created for diabetics. It is, I'm not saying it's bad for everyone. I'm saying if you want to figure out whether you got a liver or a kidney problem, that's a phenomenal way to do it. And you need to drink boatloads. Ketosis is part of this process, and we'll talk about it. It's actually part of this process. It's also part of this process, sending this one back through. You have to drink boatloads of water to keep your kidneys being able to flush out that large quantities of urea. So we'll revisit the whole protein thing like next time when I see you guys on Wednesday, right? So we'll stop there. We'll finish this set of notes and start the next one. No quiz.